just take the two numbers, subtract the largest from the smallest, and that tells you how many steps to go. So if we were changing from centimeters to millimeters here, it's 3 from 2, so just one space, and we're going to the right, so we move the decimal point to the right. So if we had 1 centimeter, how many millimeters would that be?
0.1.
No, 10.
Oh, 10.
10. Because remember, we're moving it to the right.
Okay.
So if, per se, we have 1.0 centimeters, we move it 1 to the right, so that gives us 10. Okay? Same thing in reverse if we're just going back the other way. Now, if we're going over this 0, 10 to the 0, that means that you add the numbers together. So if we said had 10 to the positive 2 and 10 to the negative 2, how many steps would that be?
4.
Yeah, 4. And then since we're moving to the right, it's always moving to the decimal to the right. If we're going to the left, it moves it to the left. And remember, if you ever have a 1, that's always equal to 1.0, so that you know where the decimal point is to move. Okay. I have to stand way over there. Come on over. Okay. First station here is temperature. Now, what was the measurement of temperature that we used on this thermometer?
Celsius.
Celsius. Okay, now whenever you do measurements on these, what's the second, let me figure out how to say this. Okay. What is the most significant figure that we can get from either in a warm water bath, cold water bath, or leave it out on the table. And then all you're going to have to do is read what it says. And right now, this one is at, you always make sure to look at it level, it's very close to 21 degrees Celsius. So, how would we do that conversion factor into Fahrenheit? If you've got your review sheet, it should be under temperature. The conversion factor is for, you take the Fahrenheit number, subtract 32 from it, and then multiply it by 5/9. And 5/9 comes out to like a 0.555556. So if you just want to do that number and multiply it by your Fahrenheit number, subtract it 32, that should work. Okay. With the triple beam balance, what's the significant figure for the triple beam balance? Well, all you have to do is look at the smallest. We've got, what are the hatch marks in between these gram marks? Well, if you look between them, there are 10 marks, so it's 1/10 of a gram. So whenever you measure these things, if this thing is, say, sitting in here, right here at the 3 and then 6 hatch marks in, that would be 3.6 grams. Okay? And we always try to measure it to the nearest 10th of a gram. Okay, now, during the test, you will not have to touch any of this stuff. It will all be set up for you already. So, now, where am I trying to get this to? Yeah, that middle line right there. So if we get it to that middle line, a bit more. And like I said, all this stuff will be set for you, so just don't touch anything during the test now. I won't have to reset anything. One more 10. Okay, so what is that set on right there? Can you read it?
469.1.
So this is right now set on 469.1. So whenever you come by later and you want to look at it. Okay, for the volume here in the graduated cylinder. Again, what would be the significant figures for this one? The hatch marks are only in 2. So again, this one is to the nearest milliliter. Now, how do you do the measurements from this one? Can I do it from here? Yeah. You have to get down to eye level. If you need to turn it a little bit, that's fine. This one is set at approximately 25 milliliters. Okay, measurement. Now, with the bones here, what they will do is they'll literally set up the station like this. You will have to measure the bone. Now, since we have this down to the nearest centimeter on here, what would be the significant figure for this one? Millimeter or to the nearest tenth of a centimeter. So, how many millimeters are in 1 centimeter? 10. So we can get it down to, actually I like to flip this around, leave it this way. Okay, do we always have to start at the end here? No. We can go through and put it anywhere on here, so long as we know where we're starting from. Like right now, I'm going to start from 20. Lean it in. So right now, this one looks to be 54.5 centimeters. And the thing that they like to do to trip you up is sometimes on the question, they won't ask for the measurement in centimeters. They'll go, okay, we want it in millimeters, or we want it in micrometers. So what you end up having to do is to take your, whatever your measurement is in centimeters, and then convert. No, and if you need practice conversion problems, there are some on the review sheet. Okay. Which side of this ruler do we use to measure with in here? Yeah, which side is the metric, top or bottom? Yeah, bottom. I've seen that mistake made so many times, so just make sure that you're using the metric on the bottom. Same thing with this one as with the large one. You can do it down to the nearest tenth of a centimeter. And they always like to put it to where these things are measured down to the tenth. So this would be 5.1 centimeters on this line. Okay, I think that's all of measurement. Does anybody have any questions about the measurements? No? All right, let's move on. Molecular models are all right here. Let's see, the easiest way to do these is just try to memorize the number of carbons and whether or not oxygens or hydrogens are associated with it. You should automatically know water. Water is the easiest one to know on here. I think water. Does that have focus?
Videotaping. No, I
Okay.
already have the PowerPoint up on Blackboard.
Okay. Methane, 1 carbon. Ethane, 2 carbons, but there's this little funky guy with the oxygen on it. That's ethanol. Remember, ethanol is our friend on the weekend. He is our enemy on the weekday. Okay, ribose and deoxyribose. Can you hold up the second one down there? Okay, easiest way to tell these apart are both of them are 5 carbon sugars. Okay, they're the only 5 carbon sugars on here. And then all you have to do after that, count the number of oxygens. Ribose has 1, 2, 3, 4, 5 oxygens on it. The deoxyribose should only have 4. And that's going to be probably the easiest way to tell these suckers apart. Glucose is going to look approximately like these two, except instead of 5 carbons, it has 6. And for that one, once you know it has 6 carbons, you don't have to guess anymore. You know that that's glucose. The last 2 on the end, remember that they're amino acids. Would you go ahead and hold the 2 of them up? You know it's an amino acid by the blue balls on there. What does the blue balls represent? Nitrogen. Okay, if you can tell, there's an NH2 on one side of the carbon, and then there's a CO2H on the other. That goes with all amino acids. It's the functional group that's kind of hanging down to the bottom that she's holding. Glycine is only going to have a hydrogen on it. Alanine has a methane group. One of these guys attached to it. Okay? And other than that, there's not much else I can help you with those. Those are just kind of memorize what they are. Let's see. Oh. Getting into the whole testing biological molecules, that is this station up here at the front. Okay. Pay very close attention to colors on these. If you'll look, there is a color difference between Biurette's and Benedict solution. The Biurette's, which is in this little bottle right here, this is what we use to test for protein. Okay? And it's a much lighter blue. I would say it's more of a sky blue. Okay? And Biurette's, whenever it changed to a positive, either turned this really deep purple color here or a pink. With the Benedicts, we were testing for sugar, and I would say that starting color, that's more of an electric blue or a turquoise blue, wouldn't you say? Okay, positive for this one was not only this reddish-orange color, but also a green color. And if it was a full-out sugar, it turned the orange color. If it was like a starch that broke down into sugars, it turned the green color. Okay, and then the last one we tested for was starch, and we used iodine. Iodine's natural color is this kind of golden brown. Okay, whenever it turns positive for starch, it's a blue-black color. Okay, so just memorize those reagents. Iodine, Benedict's, and Biurette's. Now, I wish I could take the test for you. I've had other classes ask if I would, but I'm like, I already know the material, so can't do it for you. All right. Oh, the last test, the lipid test. I almost forgot about that one. Just remember, it's whether or not the substance will evaporate. If you put water on there on the paper towel, after about 5 or 10 minutes, there's really no trace of it left. There might be a watermark, but that's it. The lipids, whether they're saturated or unsaturated, is this a saturated lipid or an unsaturated? It's vegetable oil, I'll tell you that much. Yeah, it's unsaturated because it's liquid at room temperature. If it's saturated, it's like fat. It's solid at room temperature. Remember the whole vegetable oil and bacon fat example I gave in lecture? Now, bacon fat, you can get it to liquid, but you got to heat it up first. And then as soon as you quit heating it up, it goes right back to being a solid. Oh, come on, bacon's great. Oh, I don't know, turkey bacon is good. Less fat. Okay, let's see. All right, the microscope. Okay, I'm going to tell y'all right now, this is where it always trips up everybody on the test. So for today, I'd say pay very close attention to the microscopes. Which one do they have set up for the parts? This one? Okay. All right. Come on back. Okay, let me grab one thing while you are gathering there, so I can show you how it's set up.
Somebody took pictures of
Okay.
it and put them
I'm going to show you this because this is literally how they tag stuff during the test. They'll have one of these pieces of tape cut into an arrow, right like this, and then the number will be written of the question you're supposed to answer. Okay? And then they literally, they'll go through and take it and just tape it to a part, and then they'll go, what is that part? Okay? Now, okay, do I need to go over all the parts with y'all, or do y'all kind of understand the parts? Do y'all want me to go over them again, right quick?
Sure.
Okay. Let's start from the top. These are your oculars. This is where you look into the microscope. Okay? This is binocular because it's two fields of vision. The oculars are always 10x. Okay? That means they're 10 times magnification. Working our way back, this is the tube, or body tube of the microscope. And if you can see, you can actually turn the body tube if you need to. I didn't show you all how to do that because I'm always afraid people are going to flip that off. Anyway, the back portion of it right here, this is the arm and then the base on the very bottom. Remember, one hand on the arm, one hand on the base, carry these things around. This is your plug for your light source. Okay, let's see if I can get this turned to where most of y'all can see. This is your on-off switch for your light source. Turn it around this way, light source. And then, turn it one more time, on-off switch. Okay, the knobs below it. The larger knob is your coarse adjustment knob. The smaller knob is your fine adjustment knob. On this side, this would be your fine adjustment. This side would be your coarse adjustment. Okay? Working our way down, just above the objectives here is called the nose. And then you have 4 objectives on our microscope. You have a 4x, a 10x, a 40x, and a 100x. How do you come up with the total magnification?
Multiply.
Yeah, multiply the oculars by whatever objectives on there. Now, I swear, I've seen that on the test so many times it make your ears bleed. Okay, going the rest of the way down, on the stage here, this flat portion is called the stage. This right here is called the stage clip. Okay? 2 knobs on the side. The top one is the stage adjustment knob for forward and back. The bottom knob is for right and left. Okay? Can everybody see this little lens right here at the top of the stage? This thing right here?
Yeah.
This is called your condenser. Okay? It basically takes all the light coming from the light source and focuses it into a very tight beam before it goes up through the slide. Okay, I think I've showed everybody this little adjustment lever down here just below the stage. This one right here. This is your diaphragm. This is what you use to actually either cut off or let out more light go through the condenser. Now, none of y'all have this on your microscopes. This is only in the microbiology. But there is actually a filter that goes in underneath the diaphragm. Okay? But none of y'all have it. But if they ask you what is this position here, you just say the filter. Now, I think that's all the pieces.
Which part did you say was the nose?
The nose is this little section right here, just right above the objectives. Okay, total. Okay, we kind of went over this in lecture, but they do have it in your lab book. This is considered a compound microscope because we're using 2 different lenses to do the magnification. If there was only one lens, like just the oculars, that would be a single lens microscope. They may ask you something about like scanning or transmission electron microscopes, that kind of thing. Okay. everybody has pretty much looked at these slides, all of them, right? You have at least a general idea of what they are? Because I'm thinking it'd be hard to get everybody to look into one of these microscopes, so is it okay if I just draw all this stuff on the board? Okay, let's go up to the board then. Okay, does everybody have the conversion stuff? About to erase. Does anybody need to write this down? Okay. I'll wait a second.
Let's stand right here.
Yeah, I don't want to see myself on YouTube.
That's my plan.
You know, the weird thing is I don't mind you videotaping me, but I'm always worried about how I sound.
I can post it on the discussion board and let you listen to it.
Oh, okay. It's just, every professor has their one worry in class, and I'm worried I sound goofy, or the pitch of my voice is a lot higher than I think it is. I don't know. Other professors, they just don't want to be videotaped at all. Okay. All right, with Anabaena. Anabaena is special because it's the only prokaryotic cell we looked at this semester. Okay? Whenever you're looking at it under the microscope, it literally looks like just a string of pearls that are kind of flattened or rounded. You also may see some pieces that look a little bit like this. But you're looking at this, it's about 40x to see it about that big inside the microscope. Okay? If you're looking at it at, say, 4x, it looks pretty much like this. Okay? It's just little squiggly lines. Okay? So most likely the way that they're going to show it to you is 40x, so that you can see that nice pearl shape to the strings. Okay. So let's go ahead and write this one, prokaryote. Prokaryote. Okay, all the rest of these are eukaryotes. Do y'all remember what's the difference between the two groups? Yeah, eukaryotes have all the organelles that we can think of. Prokaryotes is just basically a cell wall or a cell membrane with a little ball of DNA sitting in it. There's only one organelle that's found in both of these, and that's the ribosome. Because you have to have some way to translate that DNA into protein. All right. Okay, with the amoeba. How many of y'all actually saw the amoeba in the wet mount slide? I don't think it was anybody. Okay? Usually what they use is the stain prepared slide. Much easier to see. There's a lot more of them. But on about 4x to 10x, you should see something that looks approximately like this. Okay, with a nice large filled in dot like that. What else? Oh yeah. There should be somewhere in its body something that looks like that. Okay. Now the portions you're going to need to identify not only on the slide, but also on that model all the way back there. These little projections here are called pseudopodia. There is usually a clear circle area somewhere on the amoeba. Since the amoeba can put out the pseudopodia and move, it sometimes moves around in the cell. But this clear area is actually a contractile vacuole. I'm spelling that one? Oh. Missing the O. Okay, contractile vacuole. And then the little bitty small particles you see kind of spread out throughout the amoeba, these are food vacuums. On the model back there, it's the kind of clear circles with the 3 yellow balls sitting in them, and that's the food vacuum. The contractile vacuole is the one that's kind of larger than all the rest, and it's completely clear. Okay, now anywhere inside of the amoeba here would be considered the cytoplasm. Same thing with the model over there, just anywhere on the inside of it that's not natural organelle, that would be a cytoplasm. I think that's all the parts you need to know for the amoeba. Okay, paramecium. Again, it's the one that's back there on the absolute right-hand side of the sinks. These guys look just a little bit funny. Now, the first thing you should notice, even under the slide, is there's like a fuzzy layer all the way around the outside of the paramecium. And this is all the cilia. Sometimes the cilia can't be seen specifically because they're almost clear. So it kind of leaves like a fuzzy cellophane edge to the entire paramecium. Okay. Now, this is how I explain the oral groove and the gullet. The oral groove is the actual hole that goes into the mouth of this paramecium. The gullet is actually down inside the tube. Okay? So anything on the model that's pointed towards the outside here, that would be the oral groove. And then the gullet is way down in here where the actual food is being kind of processed in there. Okay? So on the model, this area down here, this would be considered a food vacuole. Right above it would be the gullet. And then right above that would be the oral groove. If y'all can't tell by now, I'm a very visual learner. I like drawing stuff. Okay. All right. On the model there are actual two little projections here, top and bottom on there. They're kind of whitish in color on the model. On the actual slides, they're going to look like clear areas. Both of these are, contractile vacuoles. Okay. Now, somewhere in this thing, there are two little areas. On the model, you can actually see it really well. There's this big purple blip on the model back there with the little brown one right beside it. The large one is the macro nucleus, and the small one is the micro. Okay, so, it's saying on the slide for y'all, all you have to do for the slide is the oral groove, the macronucleus, and the contractile vacuole. And that's all you should be able to identify on the slide. And then the rest of it you have to identify on the model. Any questions about the paramecium? One of these days, if I can get all y'all to draw these pictures the same way that I can and sit there and label them, I'd be a happy professor. Okay, Euglena. Now, Euglenas have this kind of funny shape to them. And if I remember correctly, the bottom edge of this thing is supposed to be more pointed. Like that. Okay, so the long, basically thread-like structures coming off the rear end of it, this is the flagellum. On the model back there, they have like 5 or 6 different small flagellum coming off the back end of the Euglena. In the slides, you may or may not be able to see it. What you usually see is basically just this section connected into the cell, but the rest of it is either too clear to see or it got ripped off whenever they were preparing the slide. Okay. Towards the back end here, right here, is the eye spot, or if it also helps you to remember it, it's the stigma. I'll accept either one of those. Okay, where's it out on the model? I just want to make sure I'm with the model. Okay, the model is showing it back here. Okay, there's a very clear area on the model, and again, this is a contractile vacuole. Okay, the nucleus, let's see. On the model, they're showing the nucleus is approximately here on this end, and usually on the slide, it's very much filled in. On the model back there, you can actually see the little squiggly pieces of DNA in it. And then around it on the model, these are the chloroplast. Now, okay, all of these pieces make sure I label all of them for you. Okay, all of these pieces you have to know on the model. Okay, for the slide, you just need to know nucleus and chloroplast. All right. Use a different color. Okay, there's only one more wet mount slide that you may see on the test, and that was the squamosal cells, squamosal epithelial cells, you know, the cheek smear. And pretty much there's just two areas you need to know on this. You need to know the, well, actually three. The cytoplasm, the plasma membrane, and then this little thing right here is the nucleus. Yeah, one of the weird things is if you take a scraping from frog cells, they don't have a nucleus. That's weird. Or, no, excuse me. Frog cells, the red blood cells, have the nucleus.
No.
And the red blood cells in our body don't. Okay, so I think that's about all the slides. We need to move over to Actually, let's do the plant cell first since we're already looking up here, and then we'll go to the animal cell in the back. Okay. Give me something to point with. Oh, okay. All right, I got a pointer. All right, plant cell. There's 3 types of plant cells we've looked at in here under the slide. I'm not going to go over all of them again. But remember in the onion cells, we weren't really looking at anything but a plant cell shape, that brick shape with the nucleus in it. Okay? The Elodiale, we were looking at the brick shape, but we were also looking for the chloroplast. Carrot cells, we're looking for, it's either a chromoplast or a carotenoid. I'll accept either one. Okay, and that's the orange squiggly lines that I drew for you. And then the potato cells, the parts of it that stain that blue-black color in the iodine, those are leucoplasts, and they contain starch. The flower petals, if you look down at them, remember how almost the entire cell was filled in with the central vacuole, and it was pink? That pink color is called anthocyanin. If you need a repeat on those, if I remember correctly, it was in section 6.3, and it was under the title Plastids. And it goes over all of those that you're supposed to be looking at. Okay, with the plant cell here, you need to know the nucleus and the nucleolus inside of it. Okay? All of this blue area outside the nucleus with the white dots on it, it's all rough ER, or rough endoplasmic reticulum. Okay? The smooth stuff, which is the dark blue here, that's the smooth ER. This large area in the center is a central vacuole. The red piece up here, and let me make sure, yeah. On the model here, you just have one, and this is the Golgi apparatus. And then all these little dots coming off of it are the seminal vesicles. The green areas here are the chloroplast, and let me check just right quick to see. No, you just need to know that this is a chloroplast. That's it. Okay, you don't have to know what the little discs are or what the little white area inside of it. Just know that the whole thing is a chloroplast. The orange ones right here, this is a mitochondria. Okay, we move out, the little holes you see here on the top, those are called plasmodesmatas. Remember, they're a type of gap junction. We talked about it during the lecture. If they point to either the holes here or these 3 green lines that they put on there, both of those are considered plasmodesmatas. And the last area is is this right here. This is considered the cell wall. And remember, the cell wall has 3 different layers to it. You've got the primary, the secondary, and then the plasma membrane on the inside. This area right here that's uncovered in blue, this is the plasma membrane. Let me just check to make sure that... Oh, and they want you to know that the little curly lines in there, for me, I'll accept either DNA, chromatin, or chromosome. All of them apply, because it's pretty much the same thing. All the little spiegly little lines in there. Okay, and I think that's just about everything for the plant cell.
So do they need to know what the layers of the cell wall?
No,
What would that be?
For-
Green, yellow, green, red is primary, secondary.
primary, secondary, and then
Middle lamella.
yeah, that's this area right here. But they don't need to know that in General Bio 1. I have them learn it in zoology and botany. Okay, so flip back. If you'll turn around, we'll go back to the animal cell. Yeah, I gotta say, with doing this class with y'all, I'm gonna really pass my test whenever I get to my PhD program. Because all the basic biology, I've taught it so many times, it's gonna be great. It's gonna be like flying through it. Okay, pretty much almost all the structures we just talked about on the plant cell are on the animal cell. Question? Oh, okay. The center area here, the whole thing is called the nucleus. And the little holes here are the nuclear pores. There are holes in the nucleus of the plant cell, they're just harder to see. Now, the nucleolus is actually this gray thing. Actually, there's two of them, here and here, in the nucleus. You see that? Right there and there. All of the little red filament stuff sitting around it is also chromatin, DNA, or chromosomes, except any of those three. All right, moving our way out. The one thing that the plants or the animal cells have that the plant cells don't have are these two structures right here. These are called the centrioles. And the centrioles are responsible for creating spindle fibers. You remember how we talked about intermediate filaments, actin filaments, and cytoskeleton in lecture? Well, most of those come from these two guys right here. Now, moving our way out, they've actually separated out. These little red dots right here and right here are just ribosomes floating in the cytoplasm. Right here and right here. But then again, you can also see those red dots on the rough ER. And again, that's what makes it rough ER. Any of these areas, they're blue and still smooth. This is smooth ER. Okay? Golgi apparatus, again, is in red, with the seminal vesicles coming right off of it. Now, the only two things that you may also need to know on this one is the yellow balls on here are lysosomes, and the purple ones are peroxisomes. Yeah, so lysosomes, peroxisomes. And then, of course, all the orange things are mitochondria.
Which one's the lysosome?
The yellow one. And see how there's several yellow ones spread out? So. And what was the purpose of the lysosomes? Yeah, they break apart just about everything. They have digestive enzymes in them that pretty much when mixed with, say, food particles, they have come in by endocytosis. If you aren't noticing, I'm kind of going over lecture tests. Okay. Anyway, the endocytosis brings in the food particles, the lysosomes mix with them, and they digest them. Peroxisomes do almost the same thing, except instead of digestive enzymes, they use like hydrogen peroxide. So, but it's pretty much the same process. Okay. The absolute last thing is what we did yesterday, and that's these three. All three of these tests here. Now they've set these up kind of like how the questions are going to be asked. Okay, like this one says, for this pH station, what is the pH of each tube? Now this is the only one that you're actually going to do yourself. During the test, you will take out 2 of the litmus paper strips. You'll go ahead and take a small drop from each test tube and go ahead and test the pH of each one. Now, from what I've just tested, you're given a pH card. Just go color to color. Test tube A would look to be about pH of 1. Test tube B looks to be a pH of 12. Now, knowing that these are heavy acids and bases, do we want to be careful during the test? No, because most likely it's going to be an acid or base in the test tube. So just be very careful moving the stuff around. Now, after you've tested, I want to always see y'all pick up these litmus paper and go ahead and drop them in this little beaker provided for you. Okay? Don't trust whoever put litmus paper before, because they might get them mixed up and you don't know which one goes with which test tube. Go ahead and run the test yourself. Now, okay. So, out of these two test tubes, since we said A was an acid and B was a base, which one of these test tubes would have the highest concentration of hydrogen ions?
Tube A.
Yeah, the acid. And that's because an acid releases hydrogen ions. Okay, buffer station. Now this is another one that kind of trips people up. If you need any help with this one, just for clarification, ask. Okay? But this one also says, which tube, A or B, is water, acid, or, excuse me. One of them is just water and acid. The other one is acid and buffer. Okay? And the way that you do this is, again, you test with the litmus strips. Okay. So out of these two pieces, which one do you think is the water and acid? Yeah, because that one is going to change pH a lot. And then you just say the other one's the base. And then that's all you do. One's water and acid, one's water, acid, and base. Okay? Or buffer. Alright, do y'all remember? I told you to memorize this name. What is this stuff? This is the pH indicator you use with the antacids.
Something purple.
Yeah, bromocresol purple. Memorize that. You're going to need to know it. It's probably going to be on the test. Now, on this one, okay, the best way to ask this is which test tube has acid in it? I'll tell you, it's a trick question. Both of them could have acid in it. But in which test tube has the buffer failed? Yeah, the yellow one. That means it has too much acid in it for the buffer to take care of it. And wherever the pH drops, that's when the bromocresol purple changes from the purple to the yellow. Now, the only other thing they want you to be able to do with something like this is if they give you the several different test tubes on there and label them like A through D, and in each one they've added one antacid and the bromocresol purple, and then they tell you they put a specific amount of acid into there. Like they'll say they put 20 drops of acid into all 4 tubes. They're going to ask you, which one's the better buffer? And how do you determine that?
Which was the least yellow?
Yeah, which one stayed purple? Because just as long as it's purple, the buffer's still working. Okay. All right, that's your entire review right there. So do y'all feel a little better going into this test now? A little bit? Okay. All I can say is that over the weekend, try to, if you can. Okay, let me ask, how do you all study?
Reviews.
Okay, the reviews, do you just take them out and read them, or read them and look them up with the book? What do you do? Just read them? Okay.
I draw pictures, I look
Write
online.
it out, like write the
Yeah.
definitions
Yeah.
out to help
That's what I do.
memorization.
Yeah.
That's what I do.
I hate to tell y'all, but just reading the review over and over again, it doesn't. Okay, most people learn 3 ways. You're either an auditory learner, you're a visual learner, or you're a kinesthetic learner. Now, if you can do stuff while you're just reading, if you read it out loud to yourself, you're getting the auditory. Then, if you can also write it or draw it out while you're reading it to yourself, you're hitting all 3, and then maybe your brain will start picking it up a little more. But just reading or just, a lot of people like to use flashcards, but I have found the act of making the flashcards usually gives people more